When an experimental therapeutic leaves the lab, it grows up in an agile, single-use clinical environment. The equipment is flexible, disposable, and scales modestly. But to supply a global market, that same biological process must be transplanted into a rigid 15,000-liter stainless steel commercial manufacturing plant. Moving from that flexible pilot scale to massive commercial infrastructure is known as tech transfer. It is commonly misunderstood as an administrative handover, a simple exchange of batch records, recipes, and quality documents from the development team to the new manufacturing site. In reality, scaling up biology introduces specific engineering conflicts. A process optimized for a small plastic bag will clash directly with the hard-piped constraints of a commercial facility. We see immediate mass balance failures, where scaled-up liquid volumes overflow existing ultrafiltration tanks, alongside aspect ratio discrepancies that alter how gases mix into the cell culture. This diagram outlines the complete sequence of bioprocessing steps, spanning from the initial cell vial thaw through upstream cell culture, downstream purification, and final fill finish. Every one of these unit operations has to be successfully mapped onto the receiving facility's hardware. This Gantt chart compares commercial tech transfer timelines on top against clinical transfers on the bottom. Clinical transfers, using disposable platforms, finish in under 3 months. Commercial transfers scale into fixed steel, demanding rigorous material sourcing, automation updates, and validation, stretching well beyond 12 months. To understand exactly what consumes that year of preparation, we are going to look at 3 diagnostic case studies: an upstream cell culture failure, a downstream routing conflict, and a drug product mechanical mismatch. Moving a process out of the lab requires redesigning biological parameters to survive the specific physical and chemical boundaries of the plant. Our first case study involves transferring a mature commercial monoclonal antibody process from a donor site's stainless steel facility to a newly constructed external contract manufacturing organization, or CMO. These graphs chart viable cell density during lab-scale shakedown runs. Across the sequential N3, N2, and N1 expansion stages, the original site's black curve and the new CMO's red curve track closely together, establishing a solid baseline of biological comparability. This graph tracks normalized titer, the actual yield of the target antibody, moving into the at-scale commercial bioreactor. Here, the curves diverge. The CMO facility suffered a sudden, unexplained 10% drop in process performance compared to historical data. To diagnose the failure, the tech transfer team used a specific investigative methodology. They removed samples from the failing commercial bioreactor immediately after inoculation and placed them into miniature lab-scale satellite models, running them in parallel with the at-scale plant. This chart maps the diagnostic results of those parallel runs. Look at the top normalized titer graph. The green line, representing the lab-scale satellites, completely overlaps the black line of the historical baseline. Meanwhile, the red line, representing the at-scale commercial run, continues to fail. Because the satellite models performed perfectly, the team could deduce that the biology and the medium preparation were sound. The failure was not caused by the facility's physical environment, its bioreactor geometry, or gassing differences at the larger scale. Ruling out the facility's physical setup shifted the investigation back to the inputs. By shipping CMO source feed materials back to the donor site for side-by-side lot comparisons, the team isolated the culprit, a specific, highly variable raw material lot within the CMO's international supply chain. When commercial scale-up fails, engineers must map the problem backward using lab-scale satellites to separate physical facility constraints from the chemistry of the inputs. Moving downstream, the engineering challenges shift from cell biology to fluid routing. In this second case study, a late-stage clinical molecule developed with non-standard technology is transferred into a heavily automated commercial facility. The legacy research and development process required the product to undergo multiple viral inactivations on separate column eluates. The commercial facility, however, was hard-piped to execute a single viral inactivation on a combined pool. Forcing the legacy sequence into the new facility would require completely severing and refabricating the plant's stainless steel pipework. To avoid millions of dollars in custom facility modifications and prolonged manufacturing downtime, the tech transfer team executed a strategy called platform conversion. This schematic details the downstream routing modifications made to align the molecule with the facility's rigid architecture. Notice the red X crossing out the harvest filters and pH adjust steps, eliminated to match existing platform piping. At column 1, the original affinity capture step required a narrow, high-resin bed. By shortening residence times and altering bed heights, engineers forced the process to fit the physical dimensions of the 2-meter columns already bolted to the floor. Adjusting the molecule to fit existing column hardware reduced overall resin costs by $1.9 million and slashed processing time for the first step from 70 hours down to 26 hours. Actively redesigning a research process to fit a commercial facility's existing physical layout is superior, both economically and temporally, to retrofitting a steel facility to accommodate a rogue process. The final manufacturing phase involves transferring the formulated drug product to an external partner for sterile filtration, aseptic filling, capping, and freeze drying. Sterile filtration presents a strict fluid dynamics challenge. The manufacturing team must ensure total homogeneity of the fluid passing through the membrane, preventing the sterile filter from diluting the excipients or absorbing the active protein. This graph maps changes in osmolality, protein concentration, and excipient percentage as a function of flush volume. By pulling sample fractions, the team identified the exact stabilization point. At 750 milliliters, the curves normalize, defining the precise flush volume required. Once the fluid is filtered and filled, the team faces the mechanical challenge of the container closure system. To meet the specific change part requirements of the CMO's automated machinery, the primary container was swapped to an ISO standard vial. This introduces a direct mechanical risk. If the capping machinery applies insufficient compression force on the stopper, the residual seal force drops. Low seal force guarantees container closure integrity failures, allowing environmental ingress and compromising the sterility of the final drug. These box plots compare residual seal force measurements. On the left, engineering runs fall dangerously below the red target threshold, indicating weak capping parameters. On the right, after machinery adjustments, the seal force is pushed cleanly above the target line, securing vial integrity. This graph overlays lyolization temperature profiles from both sites. While tuning capping force, engineers simultaneously optimize the freeze-drying cycle. The tight alignment between the new site's red profile and the donor's blue profile proves the thermal parameters successfully survived the transfer. Securing the ultimate safety of a biologic requires tuning physical and mechanical parameters, like applied capping force and thermal limits, with the same rigor applied to the upstream cell chemistry. Looking again at this project timeline, the immense weight of a commercial tech transfer becomes clear. The dense process readiness block dominating the 12 to 6-month phase is where the rigorous engineering translation happens. Upstream, readiness means qualifying international raw material supply chains to protect the cell culture from catastrophic titer drops. Downstream, readiness requires platform conversion, altering the molecule's purification parameters to obey the rigid routing and hardware limits of the commercial plant. And for the final drug product, readiness demands mechanical and physical optimization, securing container closure and fluid homogeneity. These three vectors, material qualification, platform conversion, and mechanical optimization, form the essential framework for scaling up a biotherapeutic. An acceptor facility is never a blank canvas. To scale a therapeutic, engineers treat the manufacturing plant as a rigid set of physical boundaries and re-engineer the biological process until it fits.